Welcome back, students. Now I was talking to you about how vitamin A acts at the cellular level. Now vitamin A, once it is taken up by different cells, it binds to a cellular retinol binding protein, and this will direct the retinoid to go and act wherever it is supposed to act. Now out of all this, the retinoic acid has got specific receptors on the nucleus. It acts via affecting the gene expression. So if it is retinoic acid and if this is the nucleus, retinoic acid mainly acts on the gene and it has receptors here. It acts in the form of a steroid hormone, just as the way a steroid hormone is acting, and it binds to specific retinoic acid receptor gene complex that is present in the DNA. It brings about changes in the number or changes in the translation, transcription, etc., and brings about changes at the DNA level, cellular DNA level, and thereby retinoic acid is involved in growth and differentiation. So target cells contain cellular retinoid binding proteins which directs retinoids to functional sites and protects retinoids from degradation. So retinoic acid receptor complex binds to DNA and directs the gene expression. So what are the functions of vitamin A? We found out what are the sources, what is happening, how it is transported, how it is absorbed. Now let us look at the functions of vitamin A. Minute you say vitamin A, from the time school children also know that vitamin A is involved for vision. From before government, everywhere they keep on advertising carrot for vitamin A, carotenoids, and it is involved for vision. So what is its role in vision? How does it help in vision? That is what you are supposed to know as a medical student. Now retinal turns visual light into nerve signals in the retina of the eye. Retinoic acid is required for the structural components of the eye. So one retinal and the other is retinoic acid. Both, one is to turn the visual light into nerve signals, retinoic acid for structural components of the eye. Now within the retina we have two types of cells, rods and cones. Now what do the cones do? They are responsible for vision under bright lights and they translate objects to color vision, whereas the rods are responsible for vision in dim light and translate objects to black and white vision. So in dim light you actually cannot make out which color anybody is. You can just make out that there is a figure there. It is mainly black and white vision, that is the objects are in the black and white format, especially in the dark. But you can make out so that somebody is there, what is happening in the dark light, all that you can make out. So let us look more at the functions of vitamin A. The world's visual cycle, also called as the rhodopsin cycle. Now rhodopsin is a pigment that is made up of opsin and 11-cis retinal. Opsin plus 11-cis retinal is what is present in the retina. Rhodopsin is opsin. So if I show opsin as a protein like this, so this protein is having 11-cis retinal. 11-cis retinal. Now it is important that it has to be in the cis configuration. It is also important it has to be retinal and not retinoic acid. So rhodopsin is made up of opsin plus 11-cis retinal. What happens? When light falls on the retina, this 11-cis retinal isomerizes to all-trans retinal. This immediately changes into all-trans. So what? If it changes into all-trans configuration, this will dissociate. All-trans is going to come out. The opsin is going to be liberated from this rhodopsin. Opsin comes out and all-trans retinal they separate out. Now once it separates out, there will be certain reactions that are taking place. But for those reactions to stop, it is essential that this all-trans retinal be converted to 11-cis retinal and stop those actions. Now let us see. This is what has happened minute light has fallen on the retina. Light has fallen and 11-cis retinal has been converted to all-trans retinal. Opsin has been released. So what will happen now? So what? That is the question. There occur conformational changes in the opsin portion of rhodopsin, and before it goes further, what is happening is there are a series of things that are happening. That is, first rhodopsin is converted to batho-rhodopsin is converted to lumi-rhodopsin to meta-rhodopsin 1, then to meta-rhodopsin 2, then opsin to all-trans-retinol. Did you see, just for this to dissociate, by the time it is going to dissociate into all-trans and forming opsin is released, it undergoes a series of changes, conformational changes. All these different compounds that are formed have been given a name. They are like batho, lumi, meta, etc. Meta-rhodopsin 1, meta-rhodopsin 2. So all these series changes takes place, and then finally opsin and all-trans retinal is formed. So why are these conformational changes taking place? Why so many things are happening? So when this conformational change is happening, there is activation of a G protein in the retina, and there is a change in the levels of cyclic GMP. So when this opsin is getting converted to different this, it will result in the stimulation or increasing the cyclic GMP levels. We are going at the molecular level. Cyclic GMP levels will be increased, or there will be a change in the level of cyclic GMP. When the cyclic GMP levels is going to decrease, actually, when this decreases, what happens is this causes membrane hyperpolarization and generation of a nerve impulse by closing the sodium channels. So many things are happening. When this change is happening, what will happen? It will close the sodium channels. When the sodium channels close, there will be membrane hyperpolarization. Minute there is hyperpolarization, what is happening? There is a generation of nerve impulse. So what has happened here? A visual impulse has been now converted into a nerve impulse. A visual impulse has been converted into nerve impulse in the retina. Where will this nerve impulse go? It will go to the visual cortex, and it will result in the perceiving of the object. Seeing the object is possible because of all these changes that are taking place. Because of decreased cyclic GMP, which will cause the sodium channels to close, increase the membrane hyperpolarization and go to the brain. So nerve impulse is generated, and this is transmitted to the visual cortex in the brain. Now I come to an important terminology that is known as the dark adaptation time. Minute you enter into a theater, can you see? No, it takes some time. You take some time, then you can make out where you are going in the theater, you can make out where you are, and you will be able to look at your seats and sit there. So initially when you go from a lighter, where there is lot of light, to a place where there is dark, where there is not so much light, it takes time for you to start seeing. So from color vision to black and white vision, or from the cones to the rods, it takes time for you to see. So this time by which you can start seeing is known as the dark adaptation time. You will learn more about this in physiology. I am going to talk a bit about this because we are going to talk about vitamin A here. So bright light, what it does is it depletes the stores of rhodopsin in the rods. Rods contain rhodopsin. These stores are depleted. What is rhodopsin? Opsin plus levensis retinal is rhodopsin. So when lot of light is formed, all this rhodopsin levensis retinal is converted to all-trans retinal. So it depletes the stores of rhodopsin in the rods. Cones are also there. That's why we keep on seeing during daytime. There is no problem. But during dim vision, we need rods to be active. But for that, these have been depleted. The stores of rhodopsin have depleted. So after few minutes, rhodopsin is resynthesized and vision improves. So this period is called as the dark adaptation time. Now this time, how much time it takes for you to adapt to a dark is known as the dark adaptation time. This time is increased in vitamin A deficiency. So if you look at it, one eye will contain nearly 120 million rods, each of which carries around 120 million molecules of rhodopsin. So when retinal is depleted in the retina, the opsin becomes destabilized and it is catabolized and leads to permanent destruction of the rod cell. So what is happening is, this Levensis retinal, remember it is vitamin A, it stabilizes the opsin. Suppose this Levensis retinal was not there, suppose even otherwise there is a deficiency. If there is a deficiency, Levensis retinal is not there. This opsin can get degraded, it gets destabilized, it can get degraded and it can lead to permanent rod damage. So that is the importance of vitamin A in vision, not just to convert a visual impulse into a nerve impulse, but also to stabilize the opsin portion of rhodopsin. Now what are cones? Cones are for color vision. They contain conopsin or photopsin. Now there are three types in this: cyanopsin, which is meant for blue vision, iodopsin for green, and porphyropsin for red. You will learn more about color vision in physiology. So let us stop at this level. What is happening? Now, till now I have told you all trans retinol is being formed. How is this reconverted back to Levensis retinol? One, there is an enzyme in the retina itself. In the retina itself, this can reform Levensis retinol. This is happening immediately within the retina itself. All trans retinol is converted to Levensis retinol. The other fate is it will go to the liver. It goes to the from the blood. It goes to the liver. Now in the liver, all trans retinol is converted into all trans retinol. This all trans retinol is converted to all trans retinol. Okay, retinol. Now it gets isomerized to form Levensis retinol. Levensis retinol is now then reduced to Levensis retinal, and this is transported back to the eye so that it can combine with opsin and help in rhodopsin. So one thing is, this can happen within the retina itself, or it can go to the liver and there it might be converted. Remember, retinol to retinol is just oxidation reduction reaction that is taking place. We just require NADH for this reaction to take place, retinol to retinol or retinol to retinal. So this is what is happening, and this is just an isomerase. All trans retinol to Levensis retinal is just isomerase. All to all, we just need to change it as oxidation reduction reaction that is taking place here and here. Now what are the other functions of vitamin A? Let us look at this. This is its function in vision, where all this is taking place. So Walt's visual cycle plus its conversion is taking place. Now let us look at the other functions of vitamin A. Vitamin A is involved in growth and differentiation of cells. Retinoic acid is necessary for cellular differentiation. Retinoic acid influences production, structure and function of epithelial cells that line outside that is skin, external passages that is like mucus forming cells within the body. And within the body it is involved in gastrointestinal tract, urogenital tract and the respiratory passages. So everywhere, wherever there are mucus forming cells, wherever there are epithelial cells, retinoic acid is required. So what we call it induces mucus production and it inhibits keratinization. It leads to the formation of a healthy, non-stratified, non-keratinizing epithelial layer. This is necessary. This is absolutely necessary in the GI, urogenital and respiratory passages, where it has a very important role forming of a non-stratified, non-keratinizing epithelial layer. Other functions of vitamin A, it is also involved in reproductive system. It has been shown that retinol deficiency causes miscarriages, atrophy of germinal epithelium in female rats and also causes sterility in male rats. This is mainly seen in animal studies and it is not so much seen in human beings, but it is supposed to be important for embryo development and gene expression. So overall, let us look at what is the function of retinol and retinoic acid. Retinal, AL is for vision. Retinol, OL is for reproductive system. Oic acid is for growth and differentiation of tissues, and beta carotene is acting as an antioxidant. Vitamin A also has a very important role in immunity. Why? Because its role in glycoprotein synthesis and mucus secretions, and thereby it has a role in immunity. It has also a role in iron metabolism, and retinoids also affect apoptosis or apoptosis, as some people say, and maybe it has a role in cancer chemotherapy also. Nowadays it is being explored, the role of vitamin A in cancer chemotherapy. What will happen if vitamin A is deficient? What are the causes? Decreased intake, obstructive jaundice. Why? Vitamin A requires bile salts. Cirrhosis of liver, again bile salts. Severe malnutrition, vitamin A deficiency can be seen. What will happen? The early manifestation is nyctalopia. Nyctalopia means night blindness. This is the first earliest sign. Growth retardation will be there, appetite loss, reduced taste, recurrent infection, dermatitis, dry mucous membranes. Remember, it is required for mucus forming cells, so it will result in dry mucous membrane. And one important sign that is seen is white spots, which are triangular hyperkeratotic whitish spots on the conjunctiva. This is one of the characteristic features of vitamin A deficiency. Later manifestation, this is in the early manifestation. It will lead to bone growth failure, aspermatogenesis, xerophthalmia, that is dry, thickened, lusterless eyeballs, blindness. Vitamin A is one of the easiest preventable causes of blindness. So vitamin A can just be supplemented and it will prevent blindness, and it can also lead to follicular hyperkeratosis, that is corn-like masses. Are there any therapeutic uses for vitamin A? Yes, the isotretinoin and tretinoin are used in skin disorders like acne and psoriasis. Retinoids can cause congenital malformation but not carotenoids. Okay, what is the toxicity? What will happen? The cause for toxicity could be over medication with vitamin supplements. Maybe sometimes some say topical retinoids, but if somebody is taking vitamin supplements, ingestion of polar bear liver is considered to be one of the toxic, one of the causes. This is has been asked sometimes in MCQs, which is the highest source for vitamin A, and we say that it is polar bear liver is supposed to be containing high concentrations of vitamin A. Remember, vitamin A is stored in the liver, and this is true not only for human beings but also for animals. So polar bear liver is supposed to be containing high amounts of vitamin A, and people who intake this can go into toxicity. What will be the symptoms of hypervitaminosis? Dry, itchy skin, dermatitis, fissure of lips, bone pain, edema, fatigue, renal disorders, intracranial hypertension, hemorrhages. Now, there may be because of increased activity of vitamin A on different gene expression. Remember, retinoic acid is involved in gene expression, and this might be one of the reasons for it leading to all these conditions. So with this, we come to the end of vitamin A. What are its sources? What are its effects? Do not forget its effects on vision. Vision is one of the most important functions of vitamin A. Also remember, it has got reproductive functions. Retinal, MCQ point of view, retinal is for vision. Retinol is for reproductive system. Retinoic acid is for growth and differentiation. Remember that retinoic acid acts like a steroid hormone. It goes to the nucleus, binds to the receptor there. It binds to a hormone response element, what we call as retinoic acid response element on the DNA, brings about changes in the level of transcription and translation to bring about change in the amount of protein. Which protein? Usually it could be keratin, it could be mucus producing cells, where it may be amount of mucus that is produced. So it has got a lot of role even in immunity, because it can result in the formation of mucus because of its role in mucus producing glands. It is responsible for non-keratinizing squamous epithelium formation is what is important, one of the important function of retinoic acid. So with this we come to the end of vitamin A, its sources, its functions, its toxicity. Remember, this can lead to hypervitaminosis, and it is also teratogenic. Vitamin A supplementation is not advised during pregnancy. It is actually should not be given. Retinoic acids are can cause fetal abnormalities, congenital fetal abnormalities, but carotenoids usually do not cause, the plant sources usually do not cause any congenital malformations or have any effect on the fetus. Thank you.